Thank you very much. Yes, and I'm both a professor but also a clinician, a chief clinician at Mental Health Center Copenhagen. So I both have the clinical side of the version and also the research part. And I'm happy to be invited today to the Migraine conference. And today I will talk about the lessons learned from biomarker discoveries in psychiatry. So it will also be a little bit broader than immuno-psychiatry, and will also be about our cohorts. But first, of course, in psychiatry, currently we do not have biomarkers we can use in the individual patient. So identification of biomarkers and objective markers is much needed to achieve breakthroughs in the treatment of mental disorders. And our current challenge in mental disorders and many other diseases also is that currently the choice of treatment for mental disorders is determined by trial and error using a one-size-fits-all approach, which unfortunately results in unacceptably large proportion of patients that are not responding. So if a patient comes in and has depression, we would start with the same treatment. So we do not have biomarkers where we can say this patient is more likely to respond to this given treatment than the other patients with depression. But of course the treatment is different between depression and psychosis, but these are very large groups and very likely very heterogeneous. And both schizophrenia and depression likely consist of many different subgroups with different causal mechanisms underlying their symptoms. So one of the things that we need to further improve the treatment of people with mental disorders is first of all to identify causal factors. Because if we identify causal factors, then we can also see that this patient has these causal factors, then the pharmaceutical industry can more easily identify drugs that can work against these targets. There will be precision medicine and improved treatments for these patients. But we will also look into identifying objective markers that can predict course of disease or treatment response. So even though they are not actually causal markers, if they can help us in identifying who can benefit from the given treatment or who should have increased help, it will still increase or improve the treatment of the patients. And to do some of this, we hope that we now can leverage some of the wealth of data that we have available in Denmark and also in the world, and also some of the novel data analytical approaches that are now available, so we can develop more accurate predictive models where we can integrate multiple objective measurements. Because many of these mental disorders are probably not caused by a single molecule. They are complex disorders where many very likely different organ systems are involved. Yes, so hopefully this can contribute to advancing our knowledge. But the big question is where to start. So as you can see in these pictures, there are so many places we could actually start looking for biomarkers, from genetics to transcriptomics, proteomics, metabolomics, even the microbiome, different tissues, blood, the fecal samples, and of course the brain, and deep phenotyping from clinical data or register data, and even electronic health records. And I don't have the clear answer to that, other than we are working to getting closer to the brain to actually have material that are as close to the brain as possible in deeply phenotyped cohorts, but also working on a large scale, so these can work together. But we're starting with some of the lessons that we have learned in psychiatry, is that before 2009, we didn't actually have any consistent genetic findings for mental disorders. And the first came in 2009, and the first real breakthrough for psychiatric genetics was in 2014 with this paper behind here, where they identified the first 108 GWAS significant hits for schizophrenia. But an important thing of this paper is also that these GWAS significant hits, it's not those genes that cause it in every patient. If you look at the patients with schizophrenia, it would only be fewer of these patients that have these GWAS significant hits. So the most predictive genetic marker of developing mental disorders of schizophrenia is the polygenetic risk score, and that consists for schizophrenia of 100, 2,000 different SNPs. So it's a very polygenic nature. And as you can see here on this one, this is made up in deciles, whereas this is the 10th deciles where they have 10 most of these SNPs coding for schizophrenia, and there you can see it's much more predictive. And the Danish samples are the one in the reds, where in the initial samples increased the risk of schizophrenia by 7 times, and now it's actually a bit less. But these deciles of polygenic risk scores are the ones that are currently most predictive. And that's the same pattern if you look at depression or autism or ADHD, but there are also now several GWAS significant hits, but the most predictive one is the polygenic risk scores. And from mental disorders, for this genetic field, it was also very clear that size matters, whereas from all these diseases, the first GWAS significant hits started coming when you combined the first 3,000 to 5,000 samples, and then it went up like this afterwards. So many of these consist of 100,000 of patients and controls, of course, some of them also fewer for autism. And every time there's an increased number of cases and controls in the samples, they find more hits. So another thing that it also means, when you need so large samples to identify these SNPs for the individual patients, it's not a monogenic disorder. So these SNPs have a contribution, but it's not them in themselves that are causing the whole disease. And we also see that there's okay, I have to speak louder. I'll try. We also see that there's many cross-disorder associations between the genetics of the different mental disorders. And that also comes with many of these symptoms in the individual patients are also very correlated. So many patients, if you see schizophrenia, many of the patients with schizophrenia also have what we call negative symptoms, which have an overlap with depression. And many of them also have OCD symptoms or anxiety symptoms. So many of the psychiatric disorders correlate in between. And here we did a deep learning project on one of our eyesight cohort, where we have, this is the first 80,000 that are GWASed, and then linked with the Danish registers, where we can get a family history information and a history of prior psychiatric disorders, and also birth-related factors. So if you put all of those in together with the genetics in a combined model, there you can also see, which is in the beginning of this, that the HLA region and the SNPs and even the PISs have an importance of which, if you have to distinguish between the controls and people with mental disorders, and also to distinguish in between the mental disorders. But it's less than if you just look at the family history for mental disorders. And that's actually not surprising, because the family history of mental disorders, it captures both the genetic part, but also many environmental risk factors. And what was even more important is if they had a psychiatric diagnosis of other than these 5 disorders. So if they had had prior psychiatric symptoms or a treatment, that was the most predictive, also for getting a diagnosis of schizophrenia or bipolar disorder or depression. And this is on the right. Maybe it's easier to point like this. Yeah. And this is the AOC. So you can see actually the model, this deep learning model, only on register data and genetics actually perform okay. So it has a AUC of 0.81. So it can also make ground for further work with these prediction models, where we can integrate even more data in the future, and also much more detailed biological data than is present here. And the easiest one to predict was schizophrenia, as you see here with a AUC of 0.82. And then if we move on, and this was just very briefly about the current status of the psychiatric genetics. If we look on to brain imaging studies of mental disorders, there have also the past decade have been a replication crisis in brain imaging studies of mental disorders, also due to many different methods and ways of analyzing, and many studies that were actually too small, and they didn't have a prior published study plan before making their results. So that means that many false positive findings have been published. And one of the ways that this has been solved is that the brain imaging field, they looked towards the genetic field, and so that they had this success because before in the genetic field it was the same, that there was many candidate SNPs for mental disorders that were not replicated in other studies. And then when they combined the samples, they got more consistent results that were also consistent across different cohorts. So the imaging field started doing the same, where they had made a consortium called Enigma, where they have combined many of these brain scanning studies. And these are the most recent evidence from these consortia data. And this, then again, it's very large imaging studies, where they have at least 1,000 to 5,000 individuals in the different groups. And here you can see that the red ones are schizophrenia that have the largest findings, largest differences in these brain scanning studies, particularly in the hippocampus and amygdala, whereas ADHD and depression are the most difficult ones to differentiate from healthy controls. But again, these brain imaging findings, we cannot use them in individual patients. So it's needed to have 1,000 patients that all have brain scans before we can start to look at these differences. So it's also very small differences on these standard brain imaging with functional imaging or CT scanning or MRI that have been used in these large-scale studies. And this is just a picture of the Enigma cohort that involves 50 different working groups. And they also themselves, in one of their recent papers, write that they are, of course, proud of the success they have had with this data sharing, but now there's more consistency in the finding. But one of the things that's still lacking is the same things as the genetic field is also lacking, that both these consortia only have a variable of diagnosis and then the brain imaging data, or the genetic consortia have the diagnosis and then the genetics. But they don't have much more clinical information about the cause of disease or which symptoms do they have. So I think these are also one of the future for many of these consortia, to actually combine these data with more deeply phenotyped data, so they can have it on a larger scale, and then also better distinguish different subgroups within the different diseases, and hopefully we can then get signals that are stronger than the ones that are here now. And also to do cross-disorder diagnosis, as many of these, it's not sure that the biology actually agree with the current diagnosis we have made of psychiatric disorders, as they are man-made and not based necessarily on strict biological differences. So there might also be within or across, both within the current psychiatric diagnosis, but also across different subgroups that can actually have a more targeted treatment that they get today. So yeah, so and been over the first part of this. Another part is also that the specificity is lacking in many of the identified markers. For instance, for the genetics, most of the genes involved in these polygenic risk scores are common genes that very many people have that do not have mental disorders. So they're not specific. So it's the combination of all these different SNPs that increases the risk. And also the same with the imaging studies. You can have another individual that have similar alterations as the one in mental disorders, but do not have mental disorders. So we are still lacking biomarkers that are very specific for the mental disorders that we can use in diagnosis. And also we need a larger focus on state or trait markers, because many of these prior studies have not included whether the patient have active depression or very severe depression episode at the time of the brain imaging, or if they are actually in a chronic phase or a remitted phase, which is also very likely to affect these findings. And then nowadays many people say data is gold. So a lesson from this will also be that the bigger the better, but it will also be that better samples are also needed. Better samples with much more detailed phenotypic data and biological data where we can see the interaction of many of these systems. But fortunately, in Denmark, it's a very good place to do research, as you know, where we have some good data, and we actually have nationwide Danish registers where we can follow the whole Danish population, the historical population of 9 million individuals, longitudinally, for all exposures that have required hospital contacts, for instance, infections or autoimmune diseases or birth-related factors, if you're born too early or have a low birth weight, or if your parents have a history of the disorder you look at, and then the individuals can be followed longitudinally for the risk of the given disorders. And you can also, of course, do it the other way around. And all these data is linked with prescription data also. So we have all treatments after 1995 for the whole Danish population. And that we're using also in some of the genetic data, for instance, the eyesight cohort, where I had one slide on before, where it was 135,000 that was GRAST, and then linked with these nationwide registers. Can you hear the sound okay?
Yeah.
Yeah, okay, good. And also the Copenhagen Hospital Biobank have 500,000 that are GRAST that can be linked with these registers. So in that way we can improve some of these genetic data with the phenotypic data also of the patients. And of course now we also have electronic health records to identify objective markers of disease. And we also have in Denmark one of the largest CSF biobanks, the State Serum Institute. And in our own group we have combined data from our previous trials of 2,000 individuals that are all genetically investigated and also deeply phenotyped and with blood. And then I have a new cohort we have made of Cytokines, where we both have CSF, the cerebrospinal fluid, and blood, and a microbiome in the same individuals, and a lot of phenotypical data, which we would use for biomarker discoveries. When we're looking at gene enrichment studies, and this is again from the first paper from 2014, so it has changed a bit since then. But if you see the SNPs associated with schizophrenia at that point, they actually had primarily were associated with neuronal function and synaptic functioning, and now it's a larger part that are associated with the brain. But the second biological system that were most important were actually the immune system. And also the largest genetic hit, and this was in the HLA region, as you can see up here. So there seems to also be an immune connection. So besides these things we have talked about now, it is also important to look at different biological systems. And there the immune system is a very potential promising system, which has been involved in more and more diseases lately, and also neurological diseases, and lately also psychiatric disorders. But for mental disorders, this link is actually not a new link if you look at the history. If you go back to Hippocrates, he actually back then noted a link between fever and psychotic symptoms in the patients. And at that point, they couldn't identify infections, and it was probably delirium he saw in these patients. But in 1896, one of the fathers of psychiatry, Kraepelin, he has also noted that some part of schizophrenia might actually be an immunological disease. And at that time of the point, it was due to he noticed many of his patients had skin lesions, different places in the body, but he didn't know what it was until in 1905, where the syphilis bacteria was identified. And it back then, before the area of the antibiotics, it's actually estimated that 1/3 of all patients admitted to the mental state hospitals had neurosyphilis, so infection in the brain with the syphilis bacteria causing some of these symptoms. So also the first Nobel Prize given to a psychiatrist was in 1927 to Ragnar Jalbæk, where he got it for his malaria treatment of neurosyphilis. So they didn't have antibiotics at that point, and then he induced malaria in some of the patients that activated the immune system, which fighted off the neurosyphilis in some of these patients and then made them healthy. But it was, of course, also a dangerous treatment, as some of the patients could also get very severely ill of the malaria infections. But after the Second World War, when antibiotics was brought out to the society, these patients with the neurosyphilis gradually disappeared from the mental state hospitals. And at the same period in time, the current antipsychotic and antidepressants were also introduced. So during this period, there was large advances for the treatment of mental disorders. And it was thought then that this connection between the immune system and schizophrenia was only related to syphilis, as the brain was completely protected from the immune system by the blood-brain barrier. And it was first in the beginning of the 90s that it was found out that this blood-brain barrier could become more permeable during periods with infections or inflammation or head trauma. And then later on in the 90s, it was also found out that in the brain, there are actually immune cells that were just resting, called microglia. So we didn't know that before either. And also that there are connections between the peripheral immune system and the brain, even in healthy individuals. And it's actually only a couple of years ago that it was found out that also in the brain, there's actually a lymphatic system draining the brain from immune components and toxic components for the brain. So there have been large advances in this understanding of the connection between the brain and the immune system. And several studies, which we'll go around later, have also linked the immune system to mental disorders. And first, if you look at experimental evidence from animal studies, if you into the mouse introduce LPS, this is some of the surface substance of the bacteria, which causes an inflammatory response. It will give a so-called sickness behavior with symptoms of fatigue or reduced appetite or negative mood, sleep disturbances, impaired cognition. And these are all symptoms that you can also see yourself if you just have an influenza. You can have many of these symptoms that will then disappear after the infection has cleared. And that is actually induced by the immune system's response to these infections, which also causes you to have a fever. And it's probably also made to make the body, give the body energy to actually fight off the infections. And these symptoms in animal models can be blocked by cytokine inhibitors. And this area have also received increased attention with the discovery of brain-reactive antibodies as the NMDA encephalitis, where 70% of these patients actually have their first symptoms with psychiatric symptoms, and during the course, 100% of the patients, or nearly 100%, have severe psychiatric symptoms. And also in animal studies, you can inject these anti-NMDA antibodies into the brain of the animal, and they will develop psychotic-like symptoms. But if you inject it into the periphery, nothing happens until you also inject a substance that increases the permeability of the blood-brain barrier, after which they develop the same psychotic-like symptoms. So these antibodies need to reach the brain in order to be pathogenic and also to cause neurological and psychiatric symptoms. And this disorder is very, very rare, but it responds to immunosuppressive treatments. And then when we look at the epidemiological studies, that's another angle to look at it. We here have used the nationwide Danish registers, whereas the first one, here we look at psychotic disorders. And during this, our follow-up period, we looked at 3.5 million Danish individuals. And during the period, there was 39,000 new onset of psychotic cases. And as you can see in the last blue bar, persons that had an autoimmune disease and 3 or more severe infections, they had almost 3.5 times increased risk of getting a new diagnosis with a psychotic disorder. But as you can also see on the red bars, each number of severe infections increases the risk of psychotic disorders in a dose-response associations. And these numbers are, of course, adjusted for social factors and family history of mental disorders also. And in the other graph over here, we did the same exercise, just looking at mood disorders as an outcome. And during this period, there was 91,000 new onset mood disorder cases. And here we also show that each number of infections, the red bars, increases the risk of getting a new depression diagnosis in a dose-response associations. And the last blue bar is if you have an autoimmune disease and have had 8 or more hospital contacts with infections, your risk of a new onset mood disorders is increased by over 4 times. And then when looking at infections in the brain, CNS infections, they increase the risk of, if you just look very broad at any mental disorder diagnosis, by 40%. And then if you look if there's differences between if the infection is inside the brain, encephalitis, or if it's in the meninges around the brain, we see that the risk is increased the most if the infection has been inside the brain, encephalitis, where it's increased by 81%. And actually in this study, the viral infections were associated with the highest risk. And this is a bit in contrary to what we find in the periphery, where we find that bacterial infections are associated with the highest risk of mental disorders. And that might be due to, in the periphery, bacterial infections are more likely to induce a higher immune activation that are then again more likely to actually reach the brain and can cause psychiatric symptoms. And also here we see this is a temporal association. So you see the first 6 months after you have had an infection in your brain, you have a 27 times increased risk of getting a new onset mental disorders. So this is very, very strong temporal associations for epidemiological studies. And we can actually see that the association is significantly increased even up to 20 years after. It just seemed very small compared to this large increase in the beginning. So there's clear epidemiological evidence of associations between infections and inflammation, and also with temporal associations, and that there are dose-response associations. So when looking into the genetics, immunogenetics, we did a GWAS of the susceptibility for infections to look at how much does the genetic explain when you get an infection. And from the genetics we know now, the heritability was 4%, so it actually only explained a little part. So, and it's actually not surprising that infections are mainly transmitted from person to person. So it's mainly an environmental risk factor, but there is a small genetic component, which again also was a polygenic, so we could make a polygenic risk score that predicted the risk of getting infections. But for this small genetic component, there was a moderate correlation with the genes we now know for mental disorders of 0.5. So there seemed also to be a genetic correlation between the immune system and mental disorders, which however explain a smaller part. And if we look specific at only at the HLA region, and if we do multiple testing for all the tests, we find significant association for autism and intellectual disability with the HLA region also in our Danish sample. And if we look at it over here, when it's only nominally significant results, we find many for schizophrenia also and autism. But again, it might be that there are two heterogeneous samples. So this is, if we look again in our group of individuals with mental disorders, and then only look at the ones that also have an autoimmune disease, then we find a very strong HLA signal. So there could be that there's another group that doesn't now have an autoimmune disease that are hiding in the autoimmune or in the mental disorders. So we need to combine these results with results from biomarkers, or at least Cgrp, that we can also get from the laboratory test system that we work on now, to see if we can broaden this group. And then this is also some of the first for the ICAMS study. And here we use the blood HEAL test when the child is born. So when a child is born, just on this blood HEAL test, where there's a little blood, we can make a genetic test from this. And from the genetics, this is the blue ones, we can, if you have the fourth most genes for schizophrenia, then you have a 2.3 times increased risk of developing schizophrenia when the child grows up. So in these large samples, we can use the polygenic risk score to predict how it goes with the child. And the red ones is if the child have also had an infection, then the risk is increased by 3.5 times. So the current genetics for schizophrenia and infections are additive risk factors that add on top of each other, but there were no interaction between these, at least that we could show. And another thing that is also relevant and becoming increasingly used in biomarker discoveries is a way to use genetics and perform Mendelian randomization analysis. So you can actually use the SNP to say if a potential biological marker could be causal for the disease you're studying. So you can actually use these methods to screen for biological markers in association with a disease. And of course, there should be some caution about the interpretation. And this is a study where they did this for the SNPs that code for these cytokines. Oh, disappeared. On the right, and then look with associations with schizophrenia, depression, and bipolar disorder. And here, with this Mendelian randomization analysis, find significant associations with IL-6, for instance, and schizophrenia. And I think this is a model that will come an increasing number of studies in, because you can actually make the screening very broad. But it of course only works if the marker would be directly causal. So if it's only on the causal pathway, if there's actually something else starting it, it will probably not show up in these Mendelian randomization studies. And then we're looking at moving on to the microbiome, which has also received increasing attention recent years, particularly for mental disorders, but also for many other diseases. There have just came out a meta-analysis combining all the results they could combine from 1,500 patients and 1,400 controls, and they did it cross-disorder. They looked within each mental disorder diagnosis and across the diagnosis for similarities. And the similarities they found in the patient material was that among the patients there was a decrease in microbial richness compared to controls, and there was a decrease in polygenetic diversity. And that was the main findings of the studies. But of course, when interpreting this, I think we're really lacking microbiome studies, where there's microbiome from before disease onset and also after, because many factors can influence this associations as diet and behavior, and also sampling of these materials. But still, it's interesting to follow up on. And then we're looking at the blood. Here's I showed the results from meta-analysis of studies that looked at blood levels of cytokines. And here they divided also into state and trait. So the red and the blue ones are if you have an acute relapse of first episode psychosis. So it's acute phases. And here you see that many of these cytokines are actually increased in the blood. But if you look post-treatment, which are the green ones, then they are actually decreased or not different. So at least for these cytokines, and they're of course they can change very rapid, the state of the disease is very important for biomarker discoveries. So you could actually have made these analyses only in patients that didn't have symptoms anymore, and then you wouldn't show any difference. So the likelihood of finding something is much higher when they have acute symptoms. And I guess the same would be present for most other diseases. And when we look at patients, when they come in for the first time to a mental health hospital, the first blood test they receive, they have an average CRP level of 3.5 for bipolar disorder, and 3.1 for schizophrenia, and 2.8 for depression, whereas in healthy individuals, it will be below 1 or at least around that. So there is, on average, a low-grade inflammation in the combined group of patients. And then we're looking into the CSF. We did a systematic review of all CSF studies looking at immune markers. But many of them actually looked at many different markers, so there are a few studies looking at the same markers. But this was the evidence we could compile, that there seemed to be an increased permeability of the blood-brain barrier, both in patients with depression and psychotic disorders. And they were elevated IL-6 and IL-8. And actually between 3 to 6% received a new diagnosis after a CSF test. But here we did a register-based studies on all CSF tests conducted in one of the Danish regions. So here we could also see that these basic CSF tests are not specific for mental disorders. They are actually mainly specific for CNS infections, which is not surprising. That if you have alterations in your basic CSF markers, then it's you have a very large increased risk of afterwards getting a diagnosis of CNS infections. And if you look at neurological disease, they are a little bit specific and not much other, whereas it's other markers that are used. There are specific markers for dementia and sclerosis, whereas these basic CSF markers only show a little. But for mental disorders, they are both groups have elevated risks that are quite similar, both the ones that had abnormal CSF tests, but also the ones that have received a CSF test that was within the normal range. And that could, of course, also be, if we find abnormal tests at the hospital, then you're more likely to get one of the other diagnoses, and you use the mental disorder as a rule-out diagnosis. And when going into brain imaging, looking more specific than the previous studies we looked at brain imaging. Here they had used a PET ligand against the microglia. And the first study coming out on this, up here, showed very promising results where the group with schizophrenia had increased levels of these microglia in the brain compared to healthy, and the high-risk group was in between. But subsequent meta-analysis of studies actually didn't find significant differences between these GSPO ligands in psychotic disorders, whereas in depression there were significant differences with increased activation of the microglia in the brain. And of course, here there's also a methodological problem, whereas they're very good if you have a large amount of neuroinflammation. It's very good to detect that, whereas a low grade of neuroinflammation, these methods are not currently sufficiently good. And then we're looking at post-mortem studies on CNS immune alterations. There's actually, if you take it as a group, people with schizophrenia or bipolar disorder, there's limited evidence that all of them have microglia activation and lymphocytic infiltration, as you would normally see in a neurological disease. But study indicates that there's subgroups of the patients that have particularly high immune signatures in the brain. So again, here it's not all the patients, it will be some of them having these immune alterations in the brain. And here we did meta-analysis of all treatment studies with immune modulating treatment. This is for depression. And actually, in this old placebo-controlled trial, it's almost 10,000 patients that have been included in these trials, but only 600 of them had clinical depression. But here there was an effect size of minus 0.64, which is a moderate to large effects. I've written wrong here, I can see. Whereas the one studies only looking at depressive symptoms had an effect size of minus 0.41. And in comparison, the most effective antidepressants that we have now have an effect size of 0.43. And then when doing the same exercise for psychotic disorders, there was a total of 4,000 patients included. And here we looked at mean difference in the PANSS score used to measure psychosis, and we showed a beneficial effect. But here the effect was smaller than for depression. It was a small to moderate effect. And of interest, we also show here that the larger the study was, the smaller the effect was for psychotic disorders. So in this field, there's really lacking high quality studies and also studies that take immune markers into account. Because these studies was just done on people with psychotic disorders and they were not stratified if they had elevated levels of immune markers. So also to keep this in mind, whether I said causal associations or just associations, you can use this Bradford Hill criteria for causation. And one could argue that many of these criteria are actually fulfilled within these associations between the immune system and mental disorders, with the temporality, consistency, a strong strength of associations, dose-response associations. But again, here we are lacking the specificity, because it's not, the associations seem to be infections increase almost the same, the risk for psychotic disorders as it does for depression, for instance. So also we're also thinking about, inspired by some of the studies you have done in migraine research, to having human models. Potential for mental disorders could of course be delirium or drug-induced psychiatric symptoms. And one of the best human models that are also the widely implicated is of course delirium. If you take any hospital, somatic hospital, at a given point, 1/3 of the patients will have delirium, which is an organic brain affection, which can give a variety of psychiatric symptoms, from psychotic symptoms to cognitive impairment or depressive symptoms. And these symptoms disappeared when the underlying cause is treated. And most commonly, it's infections or inflammation that cause it, but it can be many biological underpinnings to this. And also, we need to be stronger in identifying who they are, the ones with immune alterations. Because again, here, if you just look at the blood, you can see again, it's not all patients with depression, for instance, that had elevated CRP in the blood. It's actually 25% who have elevated levels of CRP, but 75% do not. So if we want to do trials targeting the immune system, we need also to use as stable markers as possible to base them on. So what we are lacking in this field is detailed immune characterization of the same individuals from the brain to the periphery with longitudinal assessment that are also cross-disorder samples. So we can actually make stratification of immune-related subgroups that we can then go back to and make randomized controlled trials targeting these specific immune alterations. And some biomarkers we have already now, but they're actually from neurology. So if you take a CSF test and they have more than 5 cells in the CSF, it is no inflammation that would likely require further investigations. Or if we detect active infection in the CSF, it would also require treatment. And of course, infections in the periphery, we will also treat that. But it would be a small group of the patients. So the question is if we can broaden this group out. And for that, we are currently using some of the samples here, that from the big data on the nationwide samples to very detailed samples. And here we have the Cytokines study, where we do CSF tests and blood and microbiome analysis at the time where they have new onset psychotic disorder or new onset depression and healthy controls. And all controls are individually matched to the patients and also for comorbidities. And we have extensive psychiatric testing and also neurological testing and for neurological soft science. And furthermore, we also have digital evaluations, recording of speech and facial recognition, and we also have wearables on, so we can make the broadest possible deep phenotyping of these patients. And it's an ongoing study where we're now taking a cut of the first 355 patients my group have included, but we will continue the inclusion to make this group, this biobank, larger and also longitudinal. And this is just some of the first results. We're just starting to analyze the samples. But here you can see the patients with depression, and it's of course important to remember that in these groups we have actually taken all out with any suspicions of immunological alterations. So we'll probably find less than we'll actually do in the whole psychiatric cohort. But here we show that CSF analyzes that patients with depression have significantly increased levels of CSF immune cells. And this is particularly present for the individuals with severe depression. And if we look at psychotic disorders, we see that they have an increased level, again, the increased amount of individuals that have CSF white blood cells above 3 compared to healthy controls. And also when we look at the CSF serum albumin ratio, we show that it's significantly increased in people with psychotic disorders, and also the CSF serum IgG ratio. But you can also see that it's significantly increased, but again, it's not all patients that will have it, it will be some of the patients, so it's a subgroup. And we will dig further into this material and also do single-cell sequencing of the immune cells to see, passing out whether it's seem to be T-cell driven or B-cell, or if it's specific subpopulation of T-cell, to also be able to do more targeted treatments. And I can see time is closing up, so but we will eventually combine all this, but they'll have metabolomics and proteomics in the same cohort of patients that will then be analyzed separately, but also in a combined deep learning models. And we are continuing with RCT trials afterwards to target these immune alterations. But I think time is up, so I talked too much, and we can just have questions for the rest. Yeah.
Thank you very much for the interesting presentation. And now it's time for questions from the audience. I see.
Hello, my name is Rune.
Please, if you can open the microphone so that we can hear you.
Can you hear me now? Can you hear me now? Hello, my name is Rune. I'm from the Danish Center. Still not?
I think you need to speak up a lot.
Hello, my name is Rune. I'm from the Danish Center. I would like to ask you a little bit regarding the imaging biomarkers, for example, from the ICAMS study. So how do you think that, for example, structural or functional imaging has helped, for example, the diagnostic practice around psychiatric disorders? So I know that, for example, you say we cannot use them to diagnose psychiatric disorders right now. So how do you think we can use them to improve the clinical practice?
Yeah, I mean, that's a good question. I think to improve right now we only use them as a differential diagnostic purposes. So if it's elderly, if a patient's above 50 years of age with the first onset mental disorder comes in, then we need to consider if a cancer, for instance, or other pathology could cause the symptoms. So there we use it. But for the main classification of psychiatric disorders, I think the scanning methods needs to be improved before they're really useful for mental disorders. So it's, I mean, higher resolution is needed, or these larger studies of the microglia, for instance. And I think currently, at least, that seems to be the way forward, because the signals in these large-scale studies is too small to use in clinical practice in psychiatry.
We have one more question.
If you're done with your question, we have a question from the online audience by Teresa Catarsi that is asking, she says it's a very interesting data, and I wonder how we consider mental illness, if it's a disease or a mental condition that does not suit our current way of living. So it's a more general question for you, I think.
Yeah. Yeah, I would say without doubt that it is a disease, but of course, I don't know if you're, in your definition, if you think it very much broader, whereas a psychiatrist, if you have schizophrenia, it's a very severe disease. I don't think there's any question of that. Individuals with schizophrenia also die 15 years earlier than the rest of the populations. And I mean, questions like that is actually the most important part that the genetics of mental disorders have contributed to, that we can clearly show in the genetics that there are genetic underpinnings that are as strong for the mental disorders as there are for diabetes and many other disorders. So I would say without doubt it is a disease, but if you can call milder symptoms that we do not diagnose as symptoms of stress or other things, then there might of course be more discussion point regarding that question.
And the question from Professor Schindler.
Yeah, Mike, thank you very much for your excellent lecture. Well, just a brief comment. I like your critiques about the imaging studies, that you need the reproducibility in these studies. We also have the same mess in migraine field and headache field. A lot of studies, some of them are small sample sizes, and these findings not always, I would say, disease specific, because you can find in any other conditions as well the same, let's say, structural changes, or sometimes also the functional changes. But my question is about the fact that we have from epidemiological studies that there is a relationship between depression and migraine. It's not like a depression causes migraine, but they kind of coexist and more frequent, vice versa. And we also have a new treatment now. It is a biologic, treatment, but it's not an immune system modulator. Cgrp receptor antagonist or anti-ligand Cgrp monoclonal antibodies. So we see from the clinical trials that those patients with the depressive symptoms, they improve also in depression, depressive symptoms. Any role of Cgrp in psychiatric disorders?
Yeah, I don't think it has been investigated enough yet, but we would very much like to look into that. I mean, that's also one of the purposes of our biobank, that we can actually look into what the association is to that, and eventually also can we see associations with the ones who get headache as an adverse event after the lumbar puncture.
After the lumbar puncture. Yeah. I know we discussed about that, but I think this aspect is very interesting because of the, we tried to also to address that by, maybe it should be interesting to study that in depression, just as a treatment for depression, see whether it works or not works. It is very speculative, why should it work? But also in the light that it's not crossing the blood-brain barrier, we also discussed about that. But maybe not all the drugs that we need for the depression should cross the blood-brain barrier.
Yeah, I think it's something that we are very open for in psychiatry, and also to do drug repurposing for other conditions. And in these meta-analyses we did in the ones sort of for the depression, many of them were actually used for trials. The ones that only had depressive symptoms were in trials of other immune conditions where they have recorded depressive symptoms. So we would actually be very interested to do the same exercises for a lot of other drugs to see if some other ones could be relevant for treatment of mental disorders.
And can you also comment on that we discussed previously about having a migraine as a different migraines, people having a migraine that might be a different signaling pathways. Do you have the same also in depression?
Yeah, I would think we have, but we have not shown it biological yet, how they actually biological distinguish from each other. The only thing we can see is that the more severe the condition is, then the more elevated biomarkers would be. For instance, peripheral CRP would be more elevated, and as we see here, the immune cells in the brain and the CSF are also more elevated if we have more severe disease. So we can use the severity, but we actually also need to look at symptoms, and there are more discussion, in particular with the immune system, into looking into neurovegetative symptoms that are maybe more immune-related, which are part of the symptoms of depression, and to distinguishing these from the other depressive symptoms. So that might also aid us.
So we have another question.
I was wondering about several of these diseases like depression, anxiety. They are episodic diseases where you are perfectly healthy in between and then you have the disease. So how much effort is spent on analyzing the same patient during and outside of depression, during and outside of anxiety, for example? I'm thinking in terms of RNA expression, metabolomics, and all this stuff here. And is there also a possibility here to do what has been incredibly productive in the migraine field, to actually study provoked attacks? I know, for example, you can provoke panic attacks quite easily with different, even with chemicals or with hypoxia and other things. That's where we might be more successful in finding biomarkers and disease and outside in the same patient.
Yeah, I very much agree, and I don't think we have done that enough in psychiatry, but particularly these experimental studies to reduce, psychosis or depression has not really been done, and I think we can particularly learn on that. So it's more observational, where as in the Cyclone study, we are including patients where they are, have a new onset depression, so they have acute disease, and then we're following them longitudinally. So So some of the patients will not have it afterwards, and then we can compare the results. But of course, we would very much like to do the same as you have done to evoke it. And it's something that I think we should actually try to investigate with the ethical committee, if we can do, because we have the same for mental disorders with several drugs where we can actually induce the symptoms.
So we have also a question online coming from Andy Cross that is asking if there's evidence that major traumatic life events can increase the inflammation in the CNS.
I think actually in the CNS it has not really been investigated, but you can see in the peripheral blood patients that had major traumatic life events, they actually have in very, very large studies have a little bit more elevated peripheral inflammation, but it's small changes, and I think some of that in those studies are also due to social factors, but they're not adjusted enough for it. But I think if you're currently stressed, the link also goes the other way around. So if you stress animals afterwards, they will have an increase in the immune response. So there's a bidirectional link between the two. So it depends on if you mean, just a past event, or if you're currently very psychologically stressed about it.
So we have 2 minutes left, so one last question from the audience.
Okay, my name is Nadia from the Danish Psychiatric Center, and it was so interesting what you presented. I'm just, I think we all agree that the distinction of psychiatric disorders and somatic disorders is quite simplistic and outdated. And then on the other hand, you say that, like in somatic disorders, where we know the cause, we call it psychiatric symptoms, but it's not a psychiatric disease. And then yet you're searching for biomarkers. And if you find biomarkers, like what is the role of the biomarker? What does it actually help you to do? Will you then shift like the diagnosis from like saying it's not psychiatry any longer, then we have a somatic disorder that we now know may cause these psychiatric symptoms, but then it's not a psychiatric disorder any longer. So the concept of biomarkers is just really challenging, and you mentioned it yourself, so maybe elaborate or comment on that. Yeah. And then I was very curious also on, you mentioned that you assess the blood-brain barrier, also in relation to migraine. Is it a peripheral or central disorder? And also in psychiatric illnesses, how did you do it? I'm just curious. You mentioned that it was decreased, the barrier. How did you measure it?
Yeah, thanks. I can start with the first question on how we measured a blood-brain barrier permeability, and of course it's not a direct measure. We used the ratio between albumin in the CSF and blood, which you can use as an estimate of the blood-brain barrier permeability, and that was increased in patients compared to controls. But of course, you can also do imaging of the blood-brain barrier, but then it has to be much larger leaks than are present in mental disorders. And we also looked at both the IgG ratio and the albumin ratio. And to take your first question, I think that's a very good question. And I hope, and I think it's also, I don't know if it's mistakes, but the psychiatry has classically been a little bit afraid of when we start to discover actually biomarkers, as we see for dementia, which was primarily psychiatric, and now when we can see it on brain imaging, then many psychiatrists has not had the will to keep it in psychiatry, and has now it's split between neurology and urology, but here in Copenhagen it's neurologists who take a bit over. So I think psychiatry also has to move in a more biological direction. But actually my own opinion is that I don't care if it's the one or the other, but we should just give the patient the best treatment. And also this distinction between neurology and psychiatry might also in the future more be it's neuroscience, and then there are different subspecialty of neuroscience.
Interesting stuff.
Thank you very much. It was very interesting.
Yeah,
and because we're out of time, if you have more questions, you can find each other over the coffee break that we have until 2:15, and then we're back for the next session. Thank you very much.
Thank you.